Hello, everyone, and welcome to this ePanel event on leveraging nanoparticle technologies for novel vaccines and therapeutics. I'm Shannon Wyman, the Scientific Communications Manager for Keystone Symposia, and I'll be your host today. Thank you for joining us. Today, we will be exploring the evolution of nanoparticle technologies from bench to bedside with field pioneers Drs. Peter Cullis, Dr. Teresa Allen, and Dr. Philip Feldner. We will examine past challenges and recent advances in nanoparticle technology and design, which are opening the door to new therapeutic modalities deploying nucleic acids to treat disease. We will discover how these fundamental breakthroughs have led to novel vaccines for COVID-19, cancer drug treatments, and gene therapy advances. In addition, we will discuss future directions in the field and potential for novel innovations in applications to further transform medicine. This event is sponsored by the VinFuture Prize, which honors exceptional minds whose breakthrough scientific research and innovations have enormous potential to improve people's everyday lives. Here to say a few words about the foundation is Dr. Lei Tai Ha, Managing Director of the VinFuture Prize, and Dr. Min An, Executive Secretary of the VinFuture Prize.
Good morning, good afternoon, and good evening, ladies and gentlemen. My name is Lei Tai Ha, and I am the Managing Director of VinFuture Prize, VinFuture Foundation. Thanks everyone for spending your time with us today. I trust that with the participation of many prominent researchers and scholars, this ePanel would bring us an insightful discussion on how technologies can support novel vaccines and therapeutics. Now, I would like to share with you some essential information about the VinFuture Foundation and VinFuture Prize. The two founders of VinFuture Foundation are Mr. Phạm Nhật Vượng and his wife, Madame Phạm Thu Hương. Mr. Vượng was born in 1968 in Vietnam, and he studied in Russia. In 2020, he was honored as the fourth Heroes of the Philanthropy in the Asia-Pacific region for his significant contribution to Vietnam's pandemic relief. He once said, "I don't care how much money I make. I want to build things that make life beautiful." The second founder is Mr. Vượng wife, Madame Phạm Thu Hương, who is the standing vice president of Vingroup. Vingroup is the largest private corporation in Vietnam, focusing on three core pillars: industry, services, and social philanthropy. We are the pioneers and the largest player in almost every market we operate in Vietnam, from real estate, hospitality, healthcare, education, to mobility. President Biden has recently praised VinFast, our electric car company, for the $4 billion investment, which expect to help create more than 7,000 jobs in North Carolina. The VinFuture Foundation was founded in December 2020 with the mission of building a future where breakthrough scientific research and technological innovations empower people and create a more equitable and sustainable world for future generations. We work toward this mission through 5 activities, as you are seeing in the slide. The first and foremost is the annual VinFuture Prize. The VinFuture Prize is the first scientific global prize from Vietnam. You know, Vietnam is still a developing country, but VinFuture Prize is one of the world's largest annual prizes in science and technology. We expect that in the years to come, Vietnam will become more visible in the global map of science and technology. There are 4 VinFuture prizes in total, one grand prize of $3 million and 3 special prizes of $500,000 each. VinFuture Prize Council has a global and diverse team of outstanding individuals from academia, research, and industry. We also have the pre-screening committee and a group of external evaluators who are all leading experts in various fields of science and technology in order to ensure that the evaluation of the prize could cover a very broad range of areas. Last but not least, here are our laureates for VinFuture Prize 2021. Professor Cullis is also joining the ePanel today. Thanks everyone for listening, and I wish you all enjoy our ePanel today. Thank you.
Hello everyone, my name is Minan and I am the Executive Secretary of the VinFuture Prize. I am very pleased to walk you through some important contents at the nomination form and the nomination portal of the VinFuture Prize. First, about the nomination portal, you can go to online.vinfutureprize.org/nomination to access the nomination portal. You can also scan the QR code you see on your screen to access the nomination portal. So on that nomination portal, you can download the instructions for nomination submission and the endorsement letter template. Inside the instructions, you can find the complete nomination form in a Word file format. And this file will help you to draft the nomination whenever you have time, and then copy paste the draft into the nomination portal. There are 7 content tabs at the nomination portal, and I would like to call your attention to tab number 5 and tab number 7. So on tab number 5, you are invited to give your evaluation of the nominee's invention, such as scientific advancement of the invention, the history of the invention, as when and how the invention started, and who else except the nominees was involved in initiating or contributing to the invention. And you are very much welcome to elaborate on most significant social economic impacts of the invention, as how this invention has transformed or will potentially transform everyday lives of millions of people. About the impacts at the VinFuture Prize. So you may know that there are 4 prize categories at the VinFuture Prize: the grand prize and the 3 special prizes for innovators from developing countries, for female innovators, and for innovators with outstanding achievements in emerging fields. For your information, the nominations for the grand prize should have a proven impact, but for the 3 special prizes, the impact could be potential, that is, in a few years. On tab number 7, about the supporting documentation and evidence, there are only 2 important documents required for a nomination form. The first one is the CV of the nominee or the nominees, and 1 endorsement letter from an expert who is not the nominator. You can also provide the endorsement at the typing space at page number 6, where you have 200 words for the endorsement. So if you face any technical issues and you have any questions, please let us know at secretariat@vinfutureprize.org. We'll be more than happy to assist you in your nomination preparation and submission, and to send you the nomination form in the Word file format so you can comfortably draft the nominations. We look forward to your nominations by 11:59 p.m. May 17, 2022, Vietnam time. And we thank you very much for your support to the success of the VinFuture Prize.
Great, and now I would like to introduce our panelists. First, we have this year's VinFuture Grand Prize winner, Dr. Peter Cullis. Welcome, Dr. Cullis.
Well, thank you.
Dr. Cullis is a professor in the Department of Biochemistry and Molecular Biology at the University of British Columbia, co-founder of Integrated Nanotherapeutics and founder of Acuitas Therapeutics, director and CEO of the Nanomedicines Innovation Network, and founding director of the Center for Drug Research and Development in Vancouver. Dr. Cullis's laboratory has made many fundamental advances in the generation, loading, and targeting of liposomal systems for delivery of conventional and genetic drugs to treat cancer and other diseases. His pioneering work in the development of lipid nanoparticles to encapsulate and protect active mRNA for delivery has enabled vaccines and therapeutics, including the COVID-19 mRNA vaccines and other life-saving medicines. Altogether, his work has opened up new possibilities to deploy nucleic acids for the prevention and treatment of many diseases in the future. Thank you for joining us today, Dr. Phyllis.
Oh, you're welcome.
I'd also like to welcome Dr. Teresa Allen to our panel today. Dr. Allen is chair of the Research Management Committee of the Nanomedicines Innovation Network, co-founder and strategic advisor for the Center for Drug Research and Development in Vancouver, visiting professor in the Nanomedicines Research Group at the University of British Columbia, and professor emeritus of pharmacology and oncology at the University of Alberta. She's also fellow of the Royal Society of Canada and fellow of the Controlled Release Society, as well as a recipient of the A.D. Bangham FRS Lifetime Achievement Award for her contributions to liposome research. Dr. Allen has been active in the drug delivery field for over 45 years and has made important contributions to the development of long-circulating liposomes and ligand-targeted nanomedicines for anti-cancer drugs and gene therapies. The product Doxil, the first anti-cancer nanomedicine approved in the world, came out of pioneering research in her laboratory at the University of Alberta. Welcome, Dr. Allen.
Thank you.
And last but not least, I would like to welcome Dr. Philip Felgner to the panel. Dr. Felgner is director of the Protein Microarray Laboratory at the School of Medicine at the University of California, Irvine, project scientist in epidemiology, and director of the Vaccine Research and Development Center at UC Irvine. He's also chair of the NIH Studies section on gene and drug delivery systems. Dr. Felgner's work is rooted in the biophysics of bilayer membranes. His pioneering work to develop lipoplexes, which are synthetic virus-like particles that deliver functional nucleic acids into cultured cells, has transformed laboratory techniques. Liposuction has since become the most widely used transfection method in vitro and even in vivo. This work spawned a separate field of nucleic acid vaccine science, which culminated in the COVID mRNA vaccines. Welcome, Dr. Felgner.
Thank you, Shannon.
We will begin today with short 15-minute presentations from each of our expert panelists about their life's work, followed by discussion and audience Q&A.
So it's a real pleasure to present to the this Keystone Symposium, and this particularly with regard to the VIN Prize, which is a truly remarkable prize, and I was honored to win it earlier on this year. So what I'm going to talk about today, and very briefly, is a journey that I've had from physics to lipid nanoparticles to gene therapy, and this has literally been gene therapy, including the Pfizer-BioNTech vaccine. It's literally been a 50-year journey. I have a number of conflicts of interest, as indicated at the bottom. Now, many people say that the mRNA vaccines are delivered in tiny bubbles of fat, and this is a little bit of a simplification. The actuality is that they are certainly very tiny. They're 100 nanometers in diameter, smaller, 100 times smaller than the typical cell, but they're really not bubbles of fats. They're rather sophisticated systems that are made of membrane lipids. And all of this started 50 years ago. I got my PhD in physics in 1972, and I really had the feeling I wanted to do something different. The most interesting problem seemed well outside the field of physics. And so I got a postdoc to go to Oxford in 1973 to study the properties of lipids found in biological membranes, really knowing very little, almost nothing about either lipids or biological membranes or anything in biology for that matter. One of the first things I did when I got to Oxford was help build an NMR machine that I then used to study properties of lipids. And why did I find membrane lipids interesting? Well, biological membranes rely on a bilayer that is composed of membrane lipids, obviously enough, that separates the inside from the outside. And without this permeability barrier, cells couldn't exist. And so membrane lipids are certainly extremely essential to life. Now, what I studied was lipid polymorphism and lipid asymmetry. Couldn't quite figure out why there were so many different, this is still true today, so many different species of lipids in membranes, and why in particular some membrane lipids were adopted non-bilayer structures. So why are those lipids there, and why do they adopt those structures? And I also got interested in lipid asymmetry, the fact that lipid composition on one side of the membrane is different than the lipid composition on the other. And I wondered if we could generate that asymmetry in response to ion gradients, and what are the consequences of these asymmetric distributions. So 50 years later, this work led to lipid nanoparticles that enable mRNA vaccines. This went through, obviously enough, a number of stages. First of all, packaging RNA, DNA into lipid nanoparticles. This is in many ways work that's still continuing on in my laboratory. The Onpattro story, which took place between in the 2000s, this was to treat a disorder known as hereditary transthyretin-induced amyloidosis, and then the COVID-19 vaccine story. So I'll very briefly just go through those events. Now, encapsulation of DNA and RNA in lipid nanoparticles requires positively charged cationic lipids for obvious reasons. You have a highly negatively charged polymer. You need a positively charged lipid to get this whole thing encased in a lipid environment or hydrophobic environment. Now, the big problem with this is that positively charged cationic lipids are highly toxic, and so we had a big issue, or how do we encapsulate nucleic acids in systems that are not highly toxic? Now, we developed an ionizable cationic lipid as part of our basic work on lipid asymmetry. And this lipid had the property that at low pH, say pH 4, the acidity of a lemon, then the lipid was positively charged, but at neutral pH, it was not charged. And so this is just indicated here. And what we found was that we could load nucleic acid polymers into lipid nanoparticles at pH 4, where they're positively charged. And then the contents were retained when we got the pH up to physiological pH values. And so we could load these lipid nanoparticles with siRNA and mRNA, and as everybody is well aware, this allows us to practice many forms of gene therapy with siRNA to inhibit production of a given protein, and messenger RNA to induce production of a particular protein. And so this is quite a wide range of possibilities in terms of therapeutics. So first we used LNP containing siRNA to treat this disorder that I mentioned, transthyretin-induced amyloidosis, a very nasty disease, as is indicated here. And this caused by production of a mutant form of transthyretin in the liver. So in a very simple hypothesis, we say, okay, if we stop production using siRNA to inhibit production of transthyretin in the liver, we should reduce the deposition of the fibrils that are caused by the mutation, which then deposit in neural tissue and in cardiac tissue. And maybe we get some stabilization, possibly recovery from the cardiomyopathies and neuropathologies that evolve. This was a spectacularly successful clinical trial. The change in neural impairment score, or the improvement, much better than placebo by values indicated here, 10 to the minus 24, 1 over Avogadro's number. There's no doubt this drug works. And in all the secondary endpoints, this was also a spectacularly successful medicine. These results were announced in 2017. So we concluded from this that Onpattro, this lipid nanoparticle formulation of siRNA, is a stable and possibly curative therapy for a previously fatal disease, a hereditary disease. This as a result, of course, of these results, then the FDA approved this drug in 2018. So we then thought, and this was a company I co-founded called Acuitas, we thought, okay, if we can deliver lipid nanoparticles containing mRNA and have a gene silenced, containing siRNA and have a gene silenced in the liver, maybe we could package mRNA and have a gene produced in the liver. And remarkably, we found we could use the liver to produce whatever protein we wanted. And this is really quite a spectacular statement to make. But to cut a long story short, we were then approached by Drew Weissman from the University of Pennsylvania and Katie Carico from BioNTech, who needed a delivery system to enable all the work that they'd done on mRNA therapeutics as vaccines, which they produced a modified mRNA to reduce immune activation and increase gene expression. And the basic thing was that we have a delivery problem. How do we get mRNA coding for viral proteins into muscle immune cells in vivo? And this work commenced, a part of it was on influenza virus, the other on Zika virus. And these results were published in 2017, showing that the lipid nanoparticle mRNA systems could produce highly effective vaccines when the mRNA coded for the Zika virus pre-membrane and envelope glycoprotein, as is indicated up here. And so this was this really gave some considerable impetus to the possibility that lipid nanoparticles containing mRNA could be excellent, could provide excellent vaccines. And so for Acuitas, this led to a collaboration with BioNTech to develop an influenza vaccine. And then in January 2020, when the pandemic hit, then all efforts, of course, switched to develop a COVID-19 vaccine that BioNTech had already been working with Pfizer on an influenza vaccine, but of course everything switched to COVID-19, and so it ended up that the lipid nanoparticle that we developed was then used to package mRNA coding for the SARS-CoV-2, the spike glycoprotein. And as we're all aware, this was again rather spectacularly successful. In November of 2020, Pfizer and BioNTech concluded a phase 3 study indicating that the vaccine was 95% effective in all age groups for preventing COVID-19 in all age groups, and ethnicities. And very well tolerated across all populations. Now, we're all well aware this has been approved in many jurisdictions now. The companies indicated they were going to produce 1.3 billion doses by the end of 2021. I think that was closer to 3 billion. And this is playing a major role in controlling the COVID-19 pandemic. But these vaccine applications are really just the tip of the iceberg. You're going to see vaccines against many more influenza, HIV, Zika, etc. The possibilities of using these types of approaches, these gene therapy approaches, for chronic diseases, cancer, heart disease, Alzheimer's, etc., is certainly in the cards. And as we saw for transthyretin-induced amyloidosis, inherited diseases, we can certainly have the potential for doing a lot for them. And so that's really the story written at a very 30,000-foot view that the development of lipid nanoparticles that enable gene therapies, including the mRNA vaccines. So it's really been quite an amazing journey, starting from very, very basic work on lipid polymorphism and lipid asymmetry. I have to acknowledge literally hundreds of collaborators in this effort, and particularly my very long-term collaborators, Mick Hope and Tom Madden, with whom I've worked for the last over 40 years. It's been quite a journey for all of us. So thank you very much.
My talk is going to be quite a personal history of where I think my laboratory has made some contributions to the field of nanomedicine. I'd like to introduce you to Dr. Alec D. Bangham, if you haven't heard of him already. He was this is a portrait of him in his house with his liposomes up here in the background. He was the first to describe multilamellar smetic mesophases, as he called them, which was termed bangosomes, but now we call them liposomes or lipid nanoparticles. He proposed that they could be used to study the functions of cell membranes and also as an artificial lung surfactant in premature babies. So that was the first commercial application for them. But before long, researchers such as Gregory Gregoriadis at University College London and Dimitri Papadopoulos at the University of California, San Francisco, and some of our colleagues were proposing that liposomes could be used for drug and vaccine delivery. Early on in my laboratory, we actually used them to study functions of cell membranes. So we used them to study membrane fusion and to reconstitute membrane proteins. And at the bottom, you'll notice that I put, I think, what are some of the key references. I want to show you this picture. It's a picture that was taken in Berlin in 2002 with myself and Dan Lassick, who was with Liposome Technology Incorporated, Alec Bangham himself, and Dimitri Papadopoulos. And this picture is notable for the fact that I'm the only one that's left alive of these three, of this group of three pioneers in the liposome field. So I rather treasure this particular photo and consider myself lucky that I've survived. In the early days, drug carriers had two major challenges that limited their clinical applications. First, they were rapidly removed into the reticuloendothelial system. And in 1987, we published the first paper that showed a way to keep liposomes for circulating longer in the body. And we initially used the monocyalloprotein GD1 to show that. And then we did quite a bit of research on the use of polyethylene glycol, which we actually now term PEG. The other problem was that drug loading was inefficient and drug leakage rates could be very high from the particles. So Doctors Deemer, Barinholtz, Cullis, and others pointed the way to efficient drug loading systems and ways that the drug leakage could be reduced from the liposomes, particularly for weak base anti-cancer drugs. And this research together led to the product Doxil, pegylated liposomal doxorubicin, which received FDA approval in 1995. And currently, the worldwide sales of Doxil are $1.25 approximately $1 million. So it's one of the poster childs of drug delivery using liposome. And there is a couple of my early publication on the long circulating liposomes containing GM1 and also containing PEG. The next challenge was demonstrating proof of concept in animal models. And then following that, finding ways to increase the selective delivery of liposomal drugs to target organs. So we demonstrated initially in an early paper in 1992 that a sustained release drug delivery system of cytarabine, soluble anti-leukemic drug, significantly reduced the toxicity and increased the lifespan in an animal model of leukemia compared to the free drug. So that was an early proof of principle for animal model applications. And then we also subsequently developed and tested a number of ligand-targeted liposomal drug delivery systems and published the first demonstration of their increased efficacy in vivo compared to non-targeted systems, where the liposomes were targeted using internalizing ligands. So something like an antibody or another type of ligand that attached to the surface of the liposomes and could trigger the liposomes being taken into the cell, so that many of these drugs that act intracellularly would be able to get into the cell and have their actions. And I've Actually, I'll just bring your attention to one paper down here from 2002 that shows that internalizing antibodies are actually necessary for the improved therapeutic efficacy of antibody-targeted liposomal drugs. In other words, the liposomes have to get inside the cell and release their drug internally. We also became interested in gene therapy quite early on. Phil Feldner had published, and of course, he'll be talking also in this symposium. He had published a number of really exciting papers demonstrating the efficacy of cationic lipid systems for transfecting cells in vitro. And we were looking for ways of increasing the efficacy of cationic liposomes relative to the gold standard of that time, which were retroviral vectors. And we wanted to deliver DNA into cells. And so over 25 years ago now, we showed that asialofetuin targeted liposomes increase the expression of a beta-galactosidase reporter gene in hepatoma cells. So that was one of our early forays into gene therapy. A lot of liposomal drugs are used for chemotherapy, and chemotherapy as it's practiced in the clinic depends on combinations of drugs with non-overlapping toxicities and side effects, non-overlapping side effects, that is. So we started looking at liposomes containing combinations of drugs and combinations of targeting agents. And so we got into trying to mimic the chemotherapy by using combinations of drugs and combinations of targeting agents. And so we were looking into a lot of models where we, like B-cell lymphomas, as you see down here, where we use combinations of doxorubicin and vincristin. We use targeting combinations of targeting agents like anti-CD20 with anti-CD19 and anti-CD22. We targeted via whole antibodies, Fab fragments and scFv fragments to see which were going to be best. And I have to say that out of those, we actually, not from my laboratory, but the first liposomal combined drug, Vexus, which contained the combination of daunorubicin and cytarabine, was approved by the FDA in 2007, and so it was reduced to commercial practice. Now, controlling the surface properties of liposomes was not only time-consuming, but difficult to scale up. So we developed methodologies for changing the surface properties of preformed liposomal drugs. So Doxil and other liposomal drugs were on the market by now. And so we were looking for ways to convert them into targeted agents. We wanted to find a technology where we could easily convert the commercial liposomal drugs to have improved properties by just inserting some ligands or antibodies or polyethylene glycol into the preformed liposomes. And so that was actually quite successful. And I have noticed that the post-insertion technology is actually being used more and more these days as a simple way of converting an already commercial liposome into having other properties like targeting ability. We also spent a lot of time doing research into the pharmacokinetics of liposomes. Now, the circulation half-life of liposomes and their biodistribution in vivo is pretty critical to their therapeutic effects and their side effects. And we performed many studies to examine the effects of size and charge and dose and route of administration and the effect of surface modifications on the pharmacokinetics of liposomes. And developed methods also to measure the degree of drug bioavailability in target cell populations like tumor cells in the body as a function of the liposome properties. And ultimately, we found that pegylated small unilamellar liposomes had a bit of a sweet spot in terms of their long circulation times over a broad dose and a broad range of sizes. And of course, Doxil fits right into that dose and size range. The other thing that we wanted to do was solve an important problem that came out of Phil Felkner's work, which was strategies to keep cationic liposomes circulating in vivo. For liposomal nucleic acids to show therapeutic effects in vivo, several important considerations have to be met. First of all, efficient entrapment of nucleic acids like mRNA in liposomes requires cationic lipids, but cationic lipids had problems with not only with increased toxicities in vivo, but also problems achieving long circulation half-lives. They ended up in the liver particularly avidly. And so we learned from our pharmacokinetic studies that positive charge can't be on the liposome surface. And we developed methods to make the surface of the liposome neutral with the positive charge and the nucleic acids trapped inside, which is really very similar to how they're making the mRNA vaccines now. And also with PEG on the surface to increase circulation times and inhibit aggregation. One thing that we did a little bit differently was that we wanted to increase the binding of the liposomes to the target cells when the target was reached. And that tended to be inhibited by PEG. And so what we did was we introduced specific internalizing targeting agents on the liposome surface to promote them to bind to the target cells and promote them to be internalized and release their contents within the target cell. And so originally we did this work and we patented it in 1998, and it was issued in 2000. And there's a reference here to where we use this method to produce a novel, long circulating and functional liposome formulation of antisense oligonucleotides targeted against MDR1 in cancer cells. So that was almost 25 years ago that we actually did that work. The patent, by the way, has now expired. We also looked for proof of principle in animal models of these targeted cationic liposomes. And we demonstrated one of the holy grails is to reach extrahepatic targets, such as solid tumors, with these cationic liposomes carrying nucleic acids. And we teamed up with a group at G. Gaslini Children's Hospital in Genoa, Italy, the group of Mirko Ponzoni. And we were able to show that cationic liposomal antisense oligonucleotide alone and given in combination with doxorubicin in a murine model of melanoma was able to increase the lifespan of the mice relative to all of the controls of with and without targeting and so forth. And also a paper that I think was really one that I liked a lot was that we along with the people in Mirko Ponzoni's group showed that cationic liposomal alkylase, siRNA, was able to increase lifespan considerably in a murine model of neuroblastoma. So basically, we got proof of principle of how to get extrahepatic targeting to solid tumors in animal models of these cationic liposomes, which is another one of those holy grails. Now, the holy grail is actually to get this happen in a human in vivo. but this sort of set the stage. So now looking to the future, my research laboratory is now closed. I closed it actually about 10 years ago, but I continue to be active in administrative positions and in doing some consulting. In the meantime, Peter Cullis and Hesse Barinholtz, Lawrence Mayer, Tom Madden, and others have been instrumental in commercializing several liposomal nanoparticle products. But the COVID vaccines using lipidic nanoparticles as carriers of COVID mRNA are seen as the most successful of all lipid particles, with sales in the billions of dollars. So this really represents the maturation of our field and opens the door to a myriad of follow-on products to treat genetic disease, rare diseases, neglected diseases, and currently incurable diseases. Thank you for your attention. That got rather small. And I just wanted, at the bottom, I have actually put a reference to a website to a conference that I'm co-organizing in June on the use of nanoparticles in the treatment of a variety of diseases looking towards the future. So thank you for your attention.
Hello everyone, I'm Phil Feldner, Director of the Vaccine R&D Center at UC Irvine in California. The title of my talk is Foundations of Vaccine and Lipid Nanoparticle Science. I'm using this timeline as an outline for my talk. Paul Ehrlich represents the period of infectious disease science that included Pasteur and Koch, when microorganisms responsible for infectious diseases were being discovered and understood. Attenuated organism technology developed then was adapted to the development of polio vaccine by Salk and Sabin, and 9 other vaccines developed by Maurice Hilleman. This was a real heyday for vaccine science. Alec Bangham is responsible for launching the liposome field. He mentored dozens of productive scientists who together were motivated by the promise of liposomal magic bullets, a term coined by Ehrlich 50 years earlier. For me, this talk begins in 1978 when I came to Tom Thompson's biophysics department in Charlottesville, Virginia, then to Syntex Research where lipofection was discovered, and Vical where Maurice Hilleman came to champion nucleic acid vaccination with us. I'll sum up with some recent COVID vaccine projects underway at UC Irvine. Here are a few things we learned in Charlottesville. We learned the exact dimensions and compositions of small unilamellar lipid vesicles. This physically stable self-assembled system is 20 nanometers in diameter and contains 2,500 lipid molecules with a combined molecular weight of 1.9 million kilodaltons. The exact dimensions of each lipid monomer in 3-dimensional space could be calculated with precision. This knowledge is useful to develop more advanced lipid nanoparticles today. The viscosity of the lipid bilayer can be a fluid like oil or a gel more like wax, depending on the chemistry and of lipid molecules, whether the aliphatic chains are saturated or unsaturated, and whether they are longer or shorter. The stability of lipid vesicles and nanoparticles is also controllable. Lipid vesicles composed of short chain lipids disintegrate rapidly in contact with biological membranes, and nanoparticles comprised of long chain lipids are stable for long periods. We also learned that the shape of lipid monomers determines the structure of self-assembled lipid nanoparticles. When the cross-sectional area of the polar head group is large compared to the aliphatic region, the cone-shaped monomers assemble into a micelle. When the space occupied by the polar head is equivalent to the aliphatic region, the cylindrical monomers form bilayers. Monomers that have a small polar head group and a large aliphatic region form inverted micelles that can organize into a hexagonal array. The mRNA vaccine lipid nanoparticles are lipid molecules contain lipid molecules with different shapes to produce a stable self-assembled structure. Some of this lipid nanoparticle biophysics was taken to Syntex research in Palo Alto. Syntex was interested in immunology and to synthesize bacterial mimetic adjuvants for better vaccines. We produced a microfluidized squalene emulsion called Syntex Adjuvant Formulation 1. I also brought the idea to make positively charged lipid molecules with the correct shape to make stable bilayer lipid vesicles. There were many potential pharmaceutical uses for positively charged vesicles. One, to make artificial virus-like particles for gene delivery. Many of us studying liposomes envisioned making synthetic virus-like particles that could deliver functional DNA and RNA into cells for gene therapy. There was a technical problem standing in the way, illustrated here. The long dimension of the DNA was much longer than the internal diameter of the typical liposome. How can we get a 5,000 base pair negatively charged DNA strand that is 1,700 nanometers long into a 70 nanometer bilayer lipid vesicle? What if we mix the cationic liposome with negatively charged DNA and capture all of the DNA into a lipid aggregate? We expected positively charged liposomes mixed with negatively charged DNA would form a visible aggregate, and we got our first surprise. They didn't produce a visible aggregate. To prove that they interact, we used sucrose density gradient centrifugation. The lipid, cationic lipid, made all of the DNA float, proving that they interact. Surprise number two, it worked right away in transfection assays. Using a very simple protocol, these lipoplexes deliver functional DNA and mRNA into cultured cells, producing the encoded protein product. Shown here is the first experiment on two different cell types. Cells produced chloramphenicol acetyl transferase enzyme after transfection of the gene. We developed a high-throughput transfection assay, screened hundreds of lipid molecules and thousands of optimized formulations to arrive at the 1987 lipofection paper in PNAS. Rick Roman, the chemist who first made the prototypical cationic lipid, DATMA, saved his lab book entry dated 7/5/84. He retired recently from Thermo Fisher and is now a cobbler making designer shoes for $5,000 a pair. What are these cationic lipid complexes we call lipoplex? The top is a negatively stained EM image showing a field of regular shaped 80 nanometer particles in a background of filaments. None of these structures are evident when either the cationic liposomes or DNA are imaged alone. Below are atomic force microscopy images showing plasmid DNA on the left, a field of 80 nanometer lipoplexes in the middle, and one lipoplex zoomed in to the right. This particular lipoplex is a condensate of a single individual 5,000 base pair plasmid. We used space-filling molecular modeling to show DATMA-dope lipids, how they organize in the presence of DNA to form the lipoplex. The modeling used the lipid packing parameters that were established by Jeff Mason and others 15 years earlier for small unilamellar liposomes and other lipid nanoparticles. It shows a 10 nanometer DNA strand in the center of an inverted micelle. According to this model, one 5,000 base pair plasmid DNA can be condensed into one 58 nanometer lipid nanoparticle surrounded by a lipid bilayer. This structure resembles mRNA vaccine lipid nanoparticles that can be imaged by cryo-electron microscopy on the next slide. On the right are 70 nanometer mRNA lipid nanoparticles. On the left is a negative stained EM of small unilamellar lipid vesicles. The vesicles on the left are empty, and the ones on the right are packed with mRNA. They can be frozen and thawed and are remarkably stable. The assembly is so good as though the RNA is a scaffold holding it together. We took some of these results to Syntex for their support, and they declined, saying gene therapy is for the year 2020. So we joined with UC San Diego scientists to start up a company called ViCal. There, in collaboration with John Wolfe from the University of Wisconsin, we got our third surprise, that DNA and messenger RNA injected intramuscularly is functional, expressing reporter genes at a level comparable to what we could get in cultured cells. The mRNA for this study was made by following methods taken from Doug Milton's two beautiful papers published in 1984, the same year that DATMA was first synthesized. Although there are many potential applications of this finding, we prioritized vaccination. With Maurice Hilleman supporting us through Merck, we demonstrated heterologous protection against influenza by immunizing with DNA encoding the nucleocapsid protein. Much clinical development in nucleic acid immunization was accomplished at ViCal, but success never reached the pinnacle that it did today, largely because of the challenging cancer and viral targets that we prioritized then, and because we didn't have the range of beautiful lipid nanoparticles that we have today. Then at UCI, we developed an approach for the discovery of vaccine antigens. We developed a protein microarray platform that can be used to make all of the individual proteins from any microorganism. The arrays are used to identify antibodies that are induced after infection. Since 2005, we made all of the proteins from 38 infectious microorganisms, printed 100,000 arrays, and probed 50,000 serum samples collected worldwide. Then in March 2020, we were prepared with a coronavirus antigen microarray that is used to measure antibodies in people who are infected with SARS-CoV-2. Here is the array containing proteins from SARS-CoV-2, SARS, MERS, common coronavirus, and influenza. The images show how the array looks when it has been exposed to serum from people who were either negative, infected with SARS-CoV-2, or vaccinated with the mRNA vaccine. The intensity of the spots increases from left to right, indicating the increase in antibody levels in the samples. Our lab was supported during the outbreak to conduct COVID research and surveillance when most of the campus was shut down. We probed 10,000 fingerstick blood specimens collected from residents throughout Orange County and from the UCI Medical Center. In December 2020, herd immunity was climbing due to the natural exposure to virus in the environment. Then, on December 16, 2020, 6,000 doses of Moderna mRNA vaccine were administered to healthcare workers at the UCI Medical Center. And the seropositivity rate in that population shot up to 78% a month later. We didn't believe the results, but in February it reached 93%, and by March it topped 99%, and we were believers. The mRNA vaccine induces spectacular immune responses. Where do we go from here? Our program in vaccine science has two research categories. Under vaccine discovery and development, we prioritize from a list of viruses, bacteria, parasites, and cancer to select vaccine antigens and test DNA, mRNA, and protein vaccines for efficacy in animal models. We need experts in all of the topics we engage. We have animal high containment facilities at UCI and have approval to conduct select agent research. In parallel, we study immunological mechanisms and lipid structure function relationships with the goal to make more effective vaccines with less reactogenicity. With that, I'd like to thank the members of my team and the laboratories of Hugh Davies and Lisa Wigar in the Vaccine R&D Center. I also want to thank our funders at DITRA, the NIH, and our corporate sponsor, Polaris Pharmaceuticals. Thank you very much.
All right, as we enter the discussion and audience Q&A portion of the event, I invite our audience to submit your questions for speakers in the Q&A tab and vote on others' questions you would like to hear answered. If your question is for a specific speaker, please say so in your question. So I'd like to begin the discussion today by asking our panelists, how did you get interested in the field in the beginning, and what did you see as its potential? And was this in contrast to what others thought at the time? So Peter, maybe you want to start.
Yeah, I in my talk I kind of illustrated how I entered the field, which was from physics. Actually, it was stimulated by a physics professor who organized a couple of seminars on biological membranes for physicists. I have to say that what I realized is that, well, A, I didn't understand anything, but B, you start to get some intimation of the complexity of biology, which usually scares the hell out of physicists. And but I found it intriguing, and so, subsequently really made an effort, obviously enough, to get into more biologically oriented things. But it's surprising how. And I always say this to students, it's a real benefit to change fields. It gives you some confidence that you can survive in an area where maybe you don't know much, but you always bring a lot to your new field by virtue of the work you've done previously. And so that's what I certainly found, is that my physics background in the end proved pretty useful. But it's not necessarily obvious that that's going to be the case. But I think it's the ability to move into new fields is really important. The multidisciplinary aspect is a very important thing to do.
Dr. Allen?
Yes, I came from an even more circuitous path. I have a PhD in oceanography, actually.
Wow.
And we were running a whole separate program where I was scuba diving all over the world and bringing back samples and looking for new anti-cancer drugs. But I did a postdoc in Miami, and we were collecting electric rays to look at the acetylcholine receptor, they hired me because I was a scuba diver, not because I was a scientist per se. And we were one of the first labs to actually isolate the acetylcholine receptor and prove that it wasn't a theoretical concept, but what it was actually a real protein. And one of the way I discovered lipids was because we wanted to reconstitute that protein into a lipid bilayer and study how it functioned. And so about that time, Alec Bangham was describing liposomes, and so we went from reconstitution and fusion experiments in the liposomes to actually using them to do drug delivery. And so it shows again that, you know, often your career takes unusual turns. And now I don't do much drug discovery from the marine environment anymore, but I've almost gone completely into the liposome area.
Wow, that's quite a jump. Very versatile. And Dr. Feldner?
Yeah, I think it was interesting that Peter and I are both biophysics. I'm not a real physics person like Peter, but I did get started in biophysics in Townshend lab. And then I got started getting recruited to go to the liposome companies, and you were being asked to make all of this practical for drug delivery. And I had no idea what to do for that. So I instead of going to the liposome companies, I went to a pharmaceutical company. And that was like going back to college because there was so much that needed to be learned there. It's kind of like Peter was saying, that you're switching fields. You know, they weren't really interested in the biophysics. They were interested in solving problems. But what happened was the biophysics that we learned really helped us to solve those problems. You know, probably helped solve problems that other people who didn't have the biophysics training wouldn't have been able to figure out how to solve.
Great. Yeah, I think it's interesting because there's often a lot of anxiety for trainees and younger students about switching fields and maybe not knowing what you're doing. But I guess scientists are pretty adaptable, and you're pretty good at using your skills to learn something new. So that's great.
Well, you know, the training shows you how to think and do research. And once you know how to do that, you can pretty much do whatever you set your mind to.
It's a great lesson. So what do you guys see as the new horizons for the applications of liposomes and nanoparticle technologies, whether it's in vaccinology, cancer immunotherapies, or gene medicines? What are the next hurdles, and how can they be applied to tackle new diseases?
Yeah, it's really I often refer to this as the third generation of pharmaceuticals. You know, we have first generation small molecules, second generation biologics, and now we have third generation gene therapies. And it's just unbelievable the range of potential applications that are coming along. They're certainly not limited to vaccines. Really, almost any disease you care to name, you can think of approaching using a siRNA, mRNA gene therapy approach. And so for younger people, you know, thinking about what would be a fun career, this is definitely, I think, an area that, but just to answer your question, that with regard to the next steps, we do need to get systems that are able to transfect tissues other than the liver. Some you know, particular cellular specificity would be good, but it might take a while to achieve that. Need to get things to the brain, etc. But all of those, we start to see ways of doing it, and it's a really exciting time.
Yeah, the sky's the limit. But the problem with prediction is because fact has a way of going its own way. Rather, separate from prediction. And so we can predict till the cows come home, but we really don't know what direction and what surprising direction that we may end up in from a past that would never have been predictable. I mean, I don't think Peter or Phil could have predicted when they were graduate students that they're or even myself that our paths would have ended up where they did. And so you have to stay open-minded, and predictions are nice, but they don't necessarily tell us anything about the future. They only tell us about the past.
Yeah, you sort of got to be in the game. I mean, my favorite example is we were busy making lipid nanoparticles containing mRNA to express proteins in the liver. We weren't thinking about vaccines at all until Drew Weissman called us up and said, "Gee, I'd really like to try your system," but that was certainly not something that we were going after. So yeah, the serendipity of science is something that, as Yogi Berra said, predictions are difficult, particularly about the future.
Yeah. In our case, just so that we have day-to-day things that we can do, we'll probably stick to vaccination. Sometimes we look at the nanoparticles and the liposomes, they trying to target them to specific tissues. They go to the liver. Peter, we were talking about this before, and it's challenging to get them to go outside the liver. But the other naturally targeted tissue happens to be the draining lymph node. That these lipid nanoparticles immediately go to after we do a typical vaccination. So it's kind of using the benefits of what is already there to get the vaccine to the perfect place to induce an immune response. But even if you're only thinking about vaccination, there's such a long agenda and different targets to prioritize, keep us busy for a long time.
Sure.
Yeah.
So we have a bunch of questions coming in from the audience, so I will ask some of those. One touches on what you were just saying, Dr. Feldner, about targeting to different tissues. Someone asked, how can nanoparticles be used to target RNAs into specific tissues upon systemic delivery? Are there any leads or ways that people are exploring to do that?
Yeah, I think that's kind of more in Peter's bailiwick there. But like I was indicating, that for vaccine applications, we already have the kind of targeting that's inherent in those lipid particles already. So we're very lucky there.
Mm-hmm.
Mm-hmm.
Yeah, Terry and I have differing opinions on this in terms of the utility of targeting ligands on the outside. What I found to be the end that works well is to take advantage of natural targeting processes such as Phil just mentioned. One of the reasons we got very lucky with the liver targeted systems was that our systems were picking up ApoE, apolipoprotein E, which triggered uptake into hepatocytes in a very efficient manner. So if you can hijack biological processes like that, it makes your job of targeting much easier. Now, whether we'll be able to do that for other tissues in an efficient way is an open question, but I do think the possibility is there.
Dr. Allen, do you have anything to add to that?
Well, we've shown it in animal models that we can get extrahepatic targeting, but that's a long way from actually demonstrating it in humans. And since my laboratory is now closed, I think that our contributions in that area are probably only going to be in the area of consulting and maybe thinking about it, but not necessarily in doing anything.
Someone else.
There's one very interesting example of targeting that's come along, and that's from Drew Weissman's lab, where he's basically doing in vivo CAR T cell therapy, and so targeting the lipid nanoparticles to T cells. In this case, he's targeted to fibrosis to cardiac therapy ends. But it's not that it's out of the question. It's just a difficult kind of technology, and in that particular case, it seems to be working very well.
One thing I'd like to point out is when you start getting into these systems where you're trying to do targeting and triggered release, and we're getting into a lot more complexity in the systems, and the more complexity you introduce into the system, the more difficult it becomes to reduce it to practice in a very practical sense from the point of view of manufacturing and scale up and things like that. And so we have to think not only in terms of what we can do in the lab, but how practical it's going to be to actually do the manufacturing and scale up as the systems become more and more complex.
Great. So we just touched on this briefly, but I want to expand a little bit more. Someone asked, can we engineer nanoparticles to target specific immune cell populations? So you mentioned the CAR T cell therapy. Are there other methods that we can use maybe to target B cells, or other ways you might imagine that these therapies might be used to target different types of immune cells?
Well, I think again, I think we're very lucky that for vaccination that the lipid nanoparticles are going into exactly the right environment to stimulate those T cells and B cells and mature the immune response. So that's already happening. But I mean, I think what we're saying is we're lucky that it's happening, but we don't know exactly how it's working. So, that's giving us a lot of opportunity to understand it better now. And, I think we have modern tools that are going to be very helpful there.
Yeah, one of the really things that we certainly don't understand yet, but is the remarkable adjuvant properties of some of these lipid nanoparticles with ionizable cationic lipids in them, and it really does account for the. I mean, you were saying, Phil, the potency is really quite amazing, and a large part of that stems from the this purely that's something we didn't know about, but the effect on the adjuvant effect, which really stimulates the immune system in a very productive manner. And seeing that we designed these systems mainly for gene expression in the liver, I think we have some ways to go here. We could probably make them even better if we start to really focus in on the the immune response aspects.
Yeah, I think so. That again, if there's anything negative about these vaccines right now, I guess you could say that most of the population has experienced the side effects for a day or two after this vaccine. So one challenge we can try to take on is to see if we can minimize those side effects while still retaining the immunogenicity. A lot of us were looking at this, and Tony Fauci was telling the world that the side effects you're getting is a good thing. Means the vaccine is working, and I think that's good. That's good advice. But it's also going to be a challenge to see whether we can retain the innate responses that are induced by the adjuvant effect of these lipid nanoparticles and reduce that side effect. It may be necessary or may not be necessary for the efficacy of the vaccine. The physicians who use this vaccine are just raving about how little toxicity, dose limiting toxicity there is, and how wonderful the vaccines are. But they can probably be made even better.
I have to laugh though, because when Dr. Fauci was saying that you know, if you get the side effects, it's proof that the vaccine was working. I've had 3 injections and I've had no side effects. And I keep thinking, oh my God, it's not working. It's not working.
Yeah, maybe it's just a sign that, you know, whatever your vaccine is working, you probably have antibodies, right? Did you?
I have no idea. But I mean, one thing, just to continue with the discussion before about targeting the immune system, one thing we have going for us is that the circulation, the blood and the lymphatic system are really, of all the tissues in the body, the most easily accessible. And so the chances are that we're going to have more luck and more success initially with targets that are more accessible, and those will include cells in the blood or lymph nodes which are bathed in lymph, because we have much easier access to those, and in the liver, of course, and spleen.
Delving a little deeper into this, the new development of the COVID-19 vaccines, were there questions about toxicity, immunogenicity, safety going in that we just didn't know? And what did the development of the vaccine teach us about the potential, and how can we use those going forward to develop better vaccines against various pathogens?
Yeah, I think it is very interesting, and Peter probably be able to weigh in on this too. You know, there were several competing companies, I guess 3 mainly, you know, BioNTech and Moderna and CureVac, and the successful ones used the less toxic, the less reactogenic, you know, the particle that produces less of a innate response, and that allows for a higher dose of the vaccine to be administered. So you're getting more messenger RNA delivered in that dose. For CureVac, they had to lower the dose to limit the toxicity, and then as a result, their performance of their vaccine didn't work up to the same level. So,
sorry,
yeah.
Phil.
Yeah, go ahead. Yeah.
No, you go ahead.
Well, so you can see how the toxicity and the efficacy can be balanced. You know, almost whenever there's a drug, any drug, the first thing that is done is you try to find the dose limiting toxicity. And then you back off from that, you know, so that you're giving the highest dose you can to show efficacy. And so it seems to me this is an example of that.
So we have to consider that in the background, we have a lot of experience with these lipid systems from Doxil, which has been received by literally millions of people over decades, with relatively few side effects. But one of the side effects that would often come up in people that received Doxil was reactions to the lipids in the system. And now data is starting to come out that some of long COVID, some of the inflammatory reactions and whatnot may be against the lipids and involving the complement system. There's been a lot of research done on that for the use of Doxil, but now I'm seeing research that's beginning to suggest that may be behind some of the reactions we're also seeing to the vaccines. The good thing about Doxil was that the reaction would occur on the first injection, but rarely on the second injection. And so we'll see whether that might prove to be the case as well with the lipid-containing COVID vaccines.
Great, Dr.
Yeah,
Phil.
there has been some you know, people point to the PEG lipid, for example, the polyethylene glycol lipid, as potentially you know, inducing anaphylactic or that kind of response. Whether that's the case or not, I think is still pretty arguable. these are things that will, you have a basic problem here that clearly the vaccine is working very well with the minimal toxicities, and those toxicities there are well controlled. If you try to introduce a new component in here, you're taking a risk. So for something that's been in billions of arms, the temptation is to say, okay, well, at least we know how this is all going to work in the general population. And so I would suspect in the vaccine area, it's going to be a little difficult to persuade somebody to put in a PEG substitute in the hope that it's going to be somewhat less, or there won't be so many of these kinds of reactions, seeing they're pretty rare. But anyway, these are things that, as we say, they'll presumably get worked out as we go down the line.
We do have an audience question actually about PEG and how the concern for drug delivery, since individuals have developed an immune response or allergy complicating that drug treatment. Is there any way we can possibly overcome this, or do you guys have any strategies or ideas where we might be able to bypass?
The thing about PEG is that the FDA puts it in a category of generally recognized as safe. And we're all ingesting PEG in our food all the time, every day, and in our cosmetics and the things we put on our face and the things we put on our mouth. PEG is actually ubiquitous in our environment. And so there are reports that there's a considerable number of people through natural exposure to PEG in our food and cosmetics who actually have antibodies against PEG. What we don't know is whether those are playing a role in the side effects that we're seeing to the vaccines, but it's certainly an interesting area to research.
Great, thank you. We also have another question about, for Peter, are there other lipids other than DODAP that can form lipid nanoparticles effectively, or is that the only one you use for your LNPs?
This is again where, how do you phrase it? That serendipity or whatever. No, we started off with DODAP, and that was just to try and get encapsulation in a system that wasn't positively charged at neutral pH. But then subsequently we found that, and so that allowed us to go in vivo, etc. But subsequently we found that by tuning up, we've synthesized, it must be 500, 600, 700 different species of ionizable cationic lipid, all of which pack in, allow us to load the nucleic acid polymers in the same way, but could have vastly different abilities to transfect cells. And so it turns out that the ionizable lipid is also playing a role in escape from the endosome, and that tuning particularly what's termed the PK of that molecule can have a very dramatic effect on the intracellular delivery. So this is again something we haven't expected. With the ionizable lipids, you can say we really did hit a bit of a gold mine, because it not only allowed us to encapsulate, but also enhance intracellular delivery, and then not only that, provides a very good adjuvant in the case of the vaccines. So it's that's been probably one of the most interesting stories here.
Great. We have another question about how else you might improve intracellular delivery of RNAs. Are there other methods beyond the ones you just discussed?
Phil, do you want to answer that?
I think it's going to come down to mechanism, understanding the mechanism better. And I think the level of screening that we're doing right now, we're checking to see if we have a vaccine that works and gives an immune response. But actually looking at the delivery of these things in more detail, I think is going to be very informative. Just to look at it in cultured cells first, that only has part of the relevancy too, because some of the things that work great in cultured cells don't work at all in vivo. But we have intravital imaging capability now, and that's going to be very informative. When you look at typically when you old-fashioned way of looking at tissues, it's from tissue sections and everything is static when you're looking at that picture. But the two-photon intravital imaging capability that we have now allows us to see that movement in real time on a television screen, and it's fantastic capability for this purpose. One of the first things that you realize is that cells are not static. They're moving around like crazy. All those T cells are constantly reaching out to try and find antigen in seconds, and they're touching each other and they're sending off calcium signals. It's a very, very dynamic process. So we haven't even begun to look at these systems in that way yet. And I think when we do, it's going to really open our eyes.
In the natural system, there's two ways of getting things into cells. The first one is it just goes through the cell membrane and gets inside, which we know for things like the nucleic acids just doesn't happen. And the other way is that for a cell to take out something is to ingest it somehow inside the cell through some kind of an internalizing mechanism. And that's the way that we're probably taking advantage of getting things into cells, because they're being endocytosed, either because we're triggering it or because it's occurring naturally. But that's not the end of the problem, is because once you get it inside the cell and it's endocytosed, it's in a little package inside the cell, and it has to be released from there to where it's going to act inside the cell. And Peter can amplify on this, I'm sure, but it seems that that's part of the key of using those charged lipids, is because they not only help it get ingested, but help it get released once it gets inside.
Yeah, and part of this, as Phil said, is really understanding the biology better. What we understand endocytosis and all of those processes to some extent, but I wouldn't say that it's a complete understanding by any means. And the more we understand mechanism, the more we'll be able to design these systems. We have theories, and sometimes those hypotheses, you can follow along in terms of trying to optimize, say, the ionizable lipids, and they're productive. But whether or not they're actually the case, I mean, what we're saying is like the ionizable lipid gets positively charged and then breaks down, the endosome, allowing the nucleic acid to escape, and that's one of our hypotheses. We've certainly never seen a production of a non-bilayer phase in an endosome. To actually capture that event would be quite unlikely. But anyway, so I do think that really this is an area of basic science that we do need to focus on.
Great. Oh, go ahead, Bill. So we probably have one more question before we're at the end of our time today. We have a question: could you adapt extracellular vesicles to deliver mRNA or plasmids for nucleic acid therapies? And if so, what would that enable you to do?
Well, yeah, extracellular vesicles have had a reputation and interest now, because if you can get them and you can use them directly for immunizations. But it is different, because they're not pure system. So I think an advantage that we have in studying mechanism is good when we have the kind of system that we can mass produce.
Yeah, they're pretty complex systems, and that's they may work at some point, but you see the complexities that come in when you're trying to use viruses as gene delivery systems. There's all kinds of issues because they're complicated. So the simple systems, keeping them as simple as you can, the non-viral systems certainly have advantages in that regard. But yeah, the exosomes of extracellular vesicles, fascinating stuff, but I'm not sure it's ready for prime time yet.
Future direction then. Speaking of future directions, what advice would you have for young scientists entering the field at this time?
Peter?
Well, okay, I'll go first. I've already said it actually, but certainly the gene therapy, this field is now, if you want to get into an rapidly growing area of therapeutics, then this is a really exciting area. As I said before, I mean, pretty much any disease is now possible to invent a medicine for in relatively simple. When you're using molecular biology, which we understand to some extent, your design of a potential therapeutic, once you know what the protein is you want to make or you want to silence or edit, then making the medicine can take, we can do this in a matter of months or less. So it's just incredible. Certainly going to get the regulatory figures are going to find this is an interesting experience, but I think for scientists, this is just a wonderful area to get into.
I'll say something. I think be open-minded, be imaginative, know what's happened in the past, but don't let it be a crutch, preventing you from thinking creatively in new directions about what might happen in the future, because quite often what we know can constrain our thinking about what we can do outside the box. So try and do some thinking outside the box, because you never know, it could be wonderful.
Very good advice.
Yeah, those are great comments. I just reiterate some of the things that we've already brought up. In this field, although it's taken so many years, one of the things that we experienced from this episode here is the continuity of science. That the vaccine development has had well over 100 years of background in it, and we're just building on that, and it's taken so many developments and so much maturation and thousands of people and scientists continuing to build on what was learned in the past. So today we're here with something. We're here with something brand new. It's now finally it's matured to this level, and it's only at the beginning of our opportunity to take advantage of it. So actually, students who are entering right now, it's a really great time, because everybody's at the starting line right now with this technology.
Great. Well, on that note, we have to wrap up, but I invite everyone to join us in the virtual networking session immediately following the event to continue these conversations and ask further questions. The remote platform allows participants to engage in small group video conversations to further discuss these topics and connect with each other. You'll find a direct link to the networking session on the closing screen and in the menu on the side of your screen. As we conclude, I would like to thank VinFuture Prize for sponsoring this event. Remember to submit your nominations for next year's VinFuture Prize before May 17th to honor those leading scientific and technological breakthroughs in your community. I would also like to thank our panelists for their time today and for their dedication to the science and research that benefits us all. It's been my privilege to speak with you. And finally, thank you to our audience for joining us today and participating in these important discussions. You can find more free ePanel events on the virtual Keystone Symposia homepage, which is part of our mission to enhance access to cutting-edge science to audiences around the world. Keystone Symposia is also hosting an upcoming conference in June on progress in vaccine development for infectious disease, which as a hybrid meeting offers live stream and on-demand options for remote viewing. We hope you will join us for another event soon. And if we can show the slide about that meeting now, that would be great. Thank you. So we hope to see you all in the networking event starting in just a moment. Thanks for joining us today.